Today we're going to be talking about cell-to-cell signaling. Specifically, we're going to talk about the signal that happens in the neurons of your eye cells. Well, what happens within the neurons. So, first we're going to start off with just a basic diagram of the eye and the light source. There's a light going into your eye. And I'm going to give you a basic eye anatomy. You should already know this. First, we have cornea, the iris, the lens, and you have the retina in the back. As light enters the eye through the cornea, through the iris, it's going to hit the back of the eye, or the retina. The retina itself is actually the axons of a ganglion cells, which is one of the few of the cell layers that compose of the retina. For the retina itself, it's composed of about 5 types of cells, but for all intents and purposes, and this example, you can just think of it in 3 major basic types of cells. You have the ganglion on top, which the axons consist of the nerve cord. Then in between the ganglion and the next group of cells, you have the bipolar cells. And then at the very back of the retina layer, you have what's called the photoreceptor cells, which is sometimes mistakenly called photoreceptors, although they are actually cells. These 3 layers, or these 3 types of cells, make up the major layers of the retina. And these are the ones that we're going to be talking about today. I'm just going to give you the quick diagram to show you which ones and their labels. Got the ganglion on the top, bipolar in the middle, then the photoreceptor cells at the bottom. So now to draw you a blown up picture of a photoreceptor cell. It's going to be the same operation for both rods and cones, but for this example we are using a rod. So inside the cell you have these layers of cells, or these layers of cell tissue, that are each surrounded by their own membrane. These layers of tissue are what's actually going to collect light. And these inner membranes are known as photoreceptor disc. Now, each one of these photoreceptor discs has a bunch of receptors on it known as rhodopsin. The rhodopsin is going to be what the actual light receptors are. It's going to be the things receiving the photons of light. And that's what we're going to concentrate on today. Rhodopsin actually belongs to a specialized family of receptors called G-protein coupled receptors, or GPCRs. They are special because they have 7 transmembrane domains, and they go coherently with the coupled G protein, which has a couple of other functions that we're going to discuss later on in the video. But right now you just need to know 7 helix receptors, 7 transmembrane domains. Now, we're going to talk about how this works. Inside the photoreceptor, you're going to have the blue part, which is the disc membrane, and then you're going to have your rhodopsin, which crosses the membrane barrier. There's your in the disc and the cytosol. And at the bottom, you're going to have your G-coupled protein, which is going to be in blue and green. The ligand for the rhodopsin is going to be retinal. These are permanently embedded into the chromophore, so they're not going anywhere. The part that is going to be moving around, causing conformational changes within the cell, is going to be transducin, which is the G protein for rhodopsin. As the photon of light hits rhodopsin, it activates the ligand retinal. There's a conformational change between the cis retinal to trans retinal. As this conformational change happens, it causes a couple of things to go on. First, we'll draw a disc again at rhodopsin, activate ligand, photon, and I'll explain that part in a minute. So now you see transducin is actually separated into 2 components. You have the alpha and the beta gamma. This works after the you have rhodopsin again, the photon, the ligand. As I was saying, G protein, transducin, and then you have GDP. So when a photon of light hits it and activates retinal, GDP is actually replaced with GTP, and it's going to cause the separation of transducin. It's going to cause it to go and interact with another protein on the membrane called phosphodiesterase, or PDE. This component of transducin is going to be the gamma, or the G alpha transducin. So again, you have the disc membrane, the side that's inside the disc, such as the cytosol. So, as transducin approaches a PDE, it has several components, two gamma, an alpha, and a beta. As it reaches of the gamma portions of PDE, you're going to have cyclic GMP be converted into GMP. Now this is going to have very important functions for the cell. But right now, again, quick overview. Alpha transducin goes to phosphodiesterase and converts cyclic GMP to GMP. Why is this important? Well, I'll explain it to you right now. Okay, you have the disc membrane, and then you have the outer cell membrane. And then you have a GMP gated channel. There's the disc, cytosol, and outside of the cell. Phosphodiesterase, and you have your sodium and calcium ions that are constantly going into the cell. And you bind down the membrane to the outer cell as cyclic GMP. This is what's going on in your eye in the dark. So there's a constant flow of sodium and calcium into the photoreceptor cell when there is no photons of light giving it any kind of response, and while cyclic GMP is bound to that gated channel. However, as cyclic GMP is converted into GMP, as in the situation in light, you're going to have less cyclic GMP available for the gated channel. The channel is going to close, effectively hyperpolarizing the cell. So basically, when you have lots of cyclic GMP, such as in the dark, your cell is constantly going to be depolarized. Where in the light, it's going to be hyperpolarized as cyclic GMP is converted into GMP. This is the signal cascade.